Hello, Namaskar. I am Ujjol Debsharma, BSc Botany Honors, 4th semester, Tufanganj Mahavidyalaya, Coach Bihar, Panchanan Borma University. So now I am presenting you a presentation about phytochrome. So here is my seminar on phytochrome. So phytochrome, here is its contents, which I will discuss in this seminar one by one. So basically, what is phytochrome? Phytochrome is a kind of photoreceptor, a pigment that plants, bacteria, and some fungi use to detect light, which regulates many developmental processes in plants. It is sensitive to light in the red and far red region of visible spectrum and can be classified as type 1 or type 2. Type 1 is activated by far red light and type 2 is activated by red light. So here you can see in this picture the absorption rate and the wavelength of light, and you can see the two form of phytochrome, PR form and PFR form. So PR form, which is type 2, is activated by absorption of red light, and PFR, which is type 1, is activated by absorbing far red light. Next is its structure. Basically, phytochrome consists of a protein covalently linked together with a light sensing built-in chromophore. So here is the structure. You can see the chromophore and its two domains. One domain contains the pigment chromophore and functions as the photoreceptor, and the other domain has protein kinase activity that triggers cellular responses. So here is the full structure of phytochrome and chromophore and its binding to the apoprotein. The chromophore is covalently linked to the protein at cysteine residue via a thioether bond. Here you can see the thioether bond. The chromophore undergoes cis trans isomerization at carbon number 15 in response to red and far red light. So here you can see the red light converts cis to trans. Next is its history and discovery. The phytochrome pigment was discovered by Starling Hendricks and Harry Borthwick at the USDA ARS Bellsville Agricultural Research Center in Maryland during a period from the late 1940s to the early 1960s. So they discovered that the red light was very effective for promoting germination or triggering flowering responses. Its distribution among tissues and cells. Phytochromes are present in most organs of plants, including roots, in etiolated seedlings. The highest phytochrome levels are usually found in meristematic regions such as bud and first node. Phytochromes are most abundant in young, undifferentiated tissues. Those cells where mRNA are most abundant, phytochromes are found there. There is a strong correlation between the abundance of phytochrome and potential of development in cells, which shows the phytochromes play an important role in controlling such developmental changes. Next thing is physiological properties of phytochrome. So the physiological nature of phytochromes can be enumerated as: number 1, phytochromes are chromoproteins comprising of a tetrapyrrole open chain of chromophore and an apoprotein. Number 2, there are two major forms of phytochrome. The PR form with absorption maxima of 667 nanometer and PFR form with absorption maxima of 730 nanometer. Number 3, the two forms of phytochromes are interconvertible. So what is interconvertible? So here you can see in this picture the two forms of phytochrome, PR and PFR. So PR form of phytochrome, when absorbs red light, it converted into PFR form, and on the other hand, in turn, PFR form converted into PR form, and PR form is blue colored and PFR form is blue green colored. So when PR, the blue colored phytochrome, absorbs the red wavelength of light, it converted into PFR, the blue green colored phytochrome. And on the other hand, in turn, PFR form of phytochrome converted into a blue colored phytochrome by absorbing far red light, and this process is called dark reversion. So the physiological role of phytochrome, number one is many flowering plants use it to regulate the time of flowering based on the length of day and night, that is called photoperiodism, we all know that, and to set circadian rhythms. Two number is it also regulates other responses including the germination of seeds, that is called photoplasty, elongation of seedlings, the size and shape and number of leaves, the synthesis of chlorophyll, and the strengthening of epicotyle or hypocotyle hoop of dicot seedlings. Number 3, it is found in the leaves of most plants. And we all know that phytochrome is found in the leaves of most plants. So here is the picture of circadian rhythms. So what is a circadian rhythm? Circadian rhythm is basically a synchronized process of plants on its various developmental stages with the environmental time. So basically it is a synchronized process of plants throughout his entire life by different stages synchronizing with environmental time by its light phase. So mechanism of action of phytochromes. So there are two types of hypotheses about this. Number one is phytochromes act as kinases and act on multiple substrates to control gene expression. And number two is phytochromes have primary reaction partners that control gene expression. So here you can see in this picture how phytochrome act as kinases and control gene expression by act on multiple substrates. So the conclusion is that phytochrome, the cytosolic photoreceptor and the signal transducing molecule, that by changing their conformation execute various physiological and photomorphogenic functions like flowering, seed germination, seedling establishment, development of photosynthetic machinery, tuberization and bud dormancy, etc. And it is also contribute to plant development by affecting plants' photosynthetic rate. So, phytochrome is very important factor of plants. So here is my brief seminar on phytochrome Hope you like it. Thank you.